Good morning, everyone. Today we will discuss a lab experiment from the second semester syllabus. That is a convex lens. So, here, what is the convex lens? This type of lens, convex lens, is thicker at the center and thinner at the edges. This type of lens is called as a convex lens. This midpoint is a focal point. The line goes from the focal point is a axial line focal. So in this type of lenses, the beam coming out from the lens, it focus at a point on two sides. For example, it is coming from this side, it focuses at all the beams focus at only one point. And if the surfaces are bent outward, the lens is called as a biconvex lens or simply convex lens. Here the rays, all the rays are focusing at single point. This point is called as a focus. This point is called as a focus. Here in the lab experiment, we have to perform one experiment per exam. In examination, you have to perform only one experiment. The question rises only one. So based on this same experiment, different kind of questions, different types of questions will rise based on the experiments. So what is the focal length here? See, in the convex lens, all the light rays focuses at a single point. That point is called as a focus. The distance between this convex lens and a focus is called as a focal length. The distance from the lens to focal point, focus, is called as a focal length. And focal power, the focal power is the reciprocal of the focal length. It is taken in meters, so focal length f represented by the small f, and focal power is represented by the capital P. So P equals to 1 by f. Here the units for the focal power is diopter. It is represented by D. So we have different types of methods to perform experiment and to find out the focal length and focal power of a given lens. Here, so first, before you entering into the experiment, you have to understand and you have to know the basic terms and definitions. Here, principal axis of a lens is the line joining the center of a curvature of the two surfaces. Here, this is the lens, these are the two surfaces. So this line is called as a principal axis, and the optical center is the point. Optical center is the point through which the ray passes undeviated through the lens. Here, for example, a ray is passing in this direction parallel to the principal axis. Then observe this. This is the glass. Here outside this is air. This is glass, and this is also air. So refractive index, this is rarer, mu1, and this is mu2, and this is also mu1. So refractive index of this lens is different from the air. So this is the principal axis. So focal point, center of the point, optical center is ray bends here. For example, you draw a normal, see here, consider this is a lens and this is a principal axis, and light rays here, if light ray is passing in this direction, then it focuses bends. It bends like this, right? And this is the point. Now this point is called as a focal focus. The distance from this lens to this focus is called as a focal length. So this from, but a ray is passing like this, it undergoes without any deviation. It undergoes without any deviation. It is called as a optical point, where the optical point, it cannot deviate its direction, but in other areas it deviates. So here observe this lens is the lens refractive index. This is a glass and here is air, say refractive index is mu1 and lens refractive index is mu2, and here air mu1. So the light ray is passing from rarer to denser. So see this is the this is a normal drawn on a surface, so it bends towards the normal. Now from denser to rarer, it is coming from glass to air, so now the normal is taken and so it comes here. So all the rays which are coming from parallel at focuses at some one point. This point is called as a focal length. But where the ray is goes without any deviation, that point is called as a optical center. That is called as a optical center. So principal focus is the point where all the rays parallel to the principal axis focuses at passing through the lens. This point is the principal focus. This point is a principal focus. The focal length is the distance between the center of the lens and focus. So here the rays are focusing at this particular point. So this point is called as a focus. The distance between lens to focus is called as a focal length, f. This is a focal length. So the focal length is a distance between the optical center and focus. If the graph cuts axis, x-axis and y-axis, then length between the origin and points of interception interrupts the graph. Here these are the basic terms involved in the this experiment. So now you have to determine the focal length and focal power of a given lens by UV method, and 1 by U 1 by V method. So you have to determine the focal length or focal power, sometimes for both the focal power and focal length by any method. So question rises like this: Find out or determine the focal length and focal power of a given lens by UV method, or determine the focal length of a given lens by UV method and plot a UV graph. And sometimes the question, based on this experiment, 5 to 6 questions will be raised. So it is determine the focal length of and focal power of a given convex lens by using UV method. That is one. Another one is determine the focal length and focal power of a given convex lens by 1 by U 1 by V method. And another one is determine the focal length of a given convex lens by UV method and plot a 1 by U 1 by V graph. And another question is determine the focal length and focal power of a given convex lens by 1 by U 1 by V method and plot a 1 by U 1 by V graph. Like this, different questions arises based on this convex lens experiment. So here, to find out the focal power, you have to find out the focal length by the which is the method asked in a question. So here we have to first we have to write in an experiment aim, apparatus, and theory, formula, and procedure, then observations and calculations, precautions, and results. So you have to follow this sequence. So here the first aim is, if he asked determine the focal length and focal power of a given convex lens by UV method, or determine the focal length and focal power of the given convex lens by 1 by U 1 by V method. So you need some apparatus to perform this experiment. So what are the apparatus you need? Here you need a convex lens, optical bench, convex lens, optical bench. This is this type of lens, and optical bench. We have an optical bench, V-shaped stand. So V-shaped stand, and a screen, and illuminated source of light, and a meter scale to measure the length between the lens and screen, or lens and object. These are the required apparatus in this experiment. So you need this illuminated light. This is a V-shaped stand, and this is lens. This is a screen to focus to get the image of the illuminated source. So here first you have to focus. First you have to determine the focal length of a given lens by distance object method. To come to a conclusion, or to know the approximate value of focal length, you have to find out the focal length by using distance object method. So what is the distance object method? Here the distance object method, the convex lens is mounted in a V-shaped stand with the principal axis horizontal. For example, see this, this is a distance object method. Sorry, this is the distance object. Consider this one is a distant object I am focusing, and this is a length which is placed in a V-shaped stand. V-shaped stand. Now focus illuminated this window, you have to get a clarity picture. The lens is focused toward the distant object such that a tall tree or any electrical pole or window through the window of the laboratory, the position of the screen is adjusted on the other side of the lens until the clear image is formed on the screen. You have to adjust the screen, you have to move the screen to get a perfect clear picture of the distance object which you are focusing. The distance between the lens, here the distance between the lens and the screen, the distance between the length and the screen directly gives the focal length. It directly gives the focal length of the object. This is the method is called as a distance object method. In distance object method, you have to focus a distance object and you have to capture the clear image of the distance object and measure the distance between the length and screen. That value, the distance, gives the focal length directly in centimeter. This is some centimeters, the distance that directly equals to focal length. This is the method to find the distance object method, the focal length of the distance object given convex lens by the distance object method. Now you have an idea what is the approximate value. You know the approximate value. For example, 24.5 is the distance between the lens and screen. The 24.5 or 25 may be the focal length of the lens. So now you have to perform experiment by using screen and source of light, illuminated source and lens. You have to arrange all these on a optical bench. For example, here this is the optical bench. Say that this is the optical bench. On optical bench, these three screen which is used to capture image, the V-shaped stand, and this is the illuminated light source. So here, see here, this is the lens placed in a V-shaped stand and illuminated light source, and this is the screen for capturing the images. So here the UV method, the distance between source and lens is called as a U, and the distance between lens and image screen is called as a V. So here convex lens is mounted on the lens stand, is placed on the optical bench containing the and the object stand which contains a wire mesh or cross wires. Here these are the cross wires kept at the some distance, that is U, from the lens. The wire mesh is illuminated to get a brighter object. The position of the screen on the other side of the lens is adjusted until the clear image is captured on a screen. If the V means the distance between this lens and image, distance of the screen and from the lens, and u be the distance of the object from the lens. Then the focal length is given by uv by u plus v. This is the formula. So here UV method, in UV method the distance, you have to take the readings u value and v value, then focal length is given by uv by u plus v in centimeter. This experiment is repeated, increasing the value of u in steps of 5 centimeter, starting from the conventional value less than the 2, so that at least 2 readings are obtained with the value of u less than the focal length 2 times of the focal length. The mean focal length f of the lens is then calculated from those uv values obtained from the observations. These values should be recorded, tabulated. Here these are the observations. For example, you have to take readings first time u is for example 20 centimeter, then v will be for example 30 centimeter. So you have to make note of these readings 1 2 3 so many times. So you have to write the readings 20 30 24 36 like this. You have to take the readings by increasing the u value. So you have taken these values and you have to take the values from this. You can take the values, if you know the u value, then you will have the 1 by u value. You have the v value, so you can find out the 1 by v value. From this you can calculate the focal length. So in 1 by f, 1 by u, you know the values, so you can substitute the values. So here the 2 graphical methods are there. One is the 1 by u, 1 by v method. For this graph 1 by u, 1 by v method, you need only 1 by uv values. But for the 1 by u, 1 by v values, you need a 1 by u, 1 by v values. So as you know the uv value, you will get the 1 by u, 1 by v simply by calculation. So you have to substitute these values. By using these values, you will draw you have to plot a graph. So see this here, the 1 by uv graph, u versus v graph is like this. So here you take a 45 degrees, then draw a line on a x-axis correspondingly. The distance from this O to x is taken as u, and O to on y-axis it is taken as v. So you can have the values. So here from this f equals to, this is OA or OB plus OC by 4. By using this formula, you can find out the focal length of a given object. Now another graph is drawn with 1 by u value and 1 by v values. 1 by u values on x-axis and 1 by v values on y-axis to the same scale here. So if the scale is, the graph is straight line. Graph is straight line when extended both the sides of the line makes equal intersections. This is OA and OB. The OA OA and OB on both axes which are equal to 1 by f. So here you can take this value f equals to 2 by OA plus OB. So f value from the graph UV method OA OB plus OC by 4 and 1 by U 1 by V method 2 by OA plus OB. You can easily find the value of interceptions on x-axis and y-axis. Then you have to substitute these values. Then you get a you will get the answer, the f value. Now, so this is the observations and calculation part. It has a more weightage. So you have to observe the values of 1 u value u1 v1, second time u2 v2, and so on. You have to take 2 or 3 or 4 or 5 different values with changing the u value. You will have, so now you have to find out the values by using this u value. You can easily find the value of 1 by u here by using v value 1 by v. From there substitute all the values 1 by u 1 by v uv values in the formula. You will get the focal length of the given lens. Then you have to find the reciprocal of the focal length, which is determined by the UV method. So for the finding out the graph, to plot a graph between 1 by u 1 by v, you need a 1 by u 1 by v values. Here to plot this 1 by u 1 by v graph, you need a 1 by u 1 by v values. To easily you can get the 1 by u 1 by v values if you know the value u and v values. So by substituting these values and plotting the graphs, you will get intercepted values on x-axis and y-axis, corresponding values. So from that graph you will get the focal length of a given lens. So calculation part is important. You have to calculate on the, otherwise you will lose the marks. So here you have to focus calculation. For example, a u value, you got the u value 30 and v value for example u value is 24, v value is 30. Then f equals to uv by u plus v. So you have to substitute this 24 into 30 by 24 plus 30, and you have to find out the focal length. You have, if you know the value of 2 into 3 is 6, then also you have to make the calculation and you have to write the answer, so you are not going to miss any small mark from this area. So right here, first you have to take calculate all the values from graph, and from the formula u and v values you have to now this is, you have the u value, you have v value. Now you have to find out the f value by calculating. You will get this, right? So now you have to take all the f's. You have to take all the f's total. You have to take the total and average value. total and average. This average gives the focal length. Average gives the focal length, right? So average focal length of a given convex lens is given, so you have to find out that is in the centimeter. So now you have to strictly follow the some precautions, otherwise you will lose the marks, and you are not able to get the correct answer if you are not following the precautions. So this is the center of the convex lens. The object and screen should be along the same horizontal line. All along the same horizontal line means here the center of the lens, object and screen should be on a same horizontal line, otherwise you are not get the exact value. The lens should be vertical in a V-shaped stand. The lens should be vertical in a V-shaped stand. For example, this is a stand, so lens will be vertical. You have to put the lens. You have to put the lens vertically, right? So reading should be taken without any parallax. Reading should be taken without any parallax. The distance should be more than the focal length. The distance of the object, the distance of the object should be more than, the distance of the object should be more than 2f. As we know the f value by distance object method. We know this value by distance object method, so it should be more than the 2f. The upright supporting of the optical element should be rigid and mounted vertically. Whatever the whatever the optical instruments supporting of the optical elements should be rigid and mounted vertically. The aperture of the lens should be small, otherwise the image formed will not be distinct. It is not clear. I should be placed at the distance more than the 25 centimeter of the image needle. So if you are using the illuminated nest or illuminated wires, or you are using illuminated needle, then our eye should be away 25 centimeter away from the needle. An error may be rise in the observation if the top of the optical bench is not horizontal. If the optical bench is not horizontal, not flat, then error may be rises. And similarly, if the tip of the pin or the wire, illuminated wire and optical center of the lens are not at the same horizontal level, then some error will arises. The general instructions should be followed in all optical bench experiments. Must be taken care of. All the general instructions which are given in all the optical bench experiments must be taken care of, otherwise the errors will occur. The corrected value of the distances u and v must be put in the formula for calculating f, and then the mean value of f should be taken. You have to note down the correct u and v values in the tabular form, and you have to substitute those values u and v values correctly and calculate to get the f value. The f value must not be made with using the mean values of u and v. Some of the students are making wrong while calculating the f value. you have to take the mean of f value, not the don't take the mean of u value and mean of v value. You have to take only the here some of the students are taking total and mean of the u and mean of the v. So this is wrong one. You have to take u value, v value, and you have to find out the f. Second time u2, v2 and f2, so on. You have to take the mean of only f. You have to take mean of f only, f1, f2, f2 mean value, not the mean of u or mean of v. So take care about this one, otherwise you will get wrong answer. And finally you have to write the result. So the result should be written directly and in a smaller manner. No need to explain all the thing, just you have to write a single line answer. What is your question? For example, your question is determine the focal length of a given convex lens. So your reading your answer result must be describes only, it shows only the focal length of a given convex lens is f equals to some 20 centimeter or 25 centimeter. Don't elaborate the answer. So here the result is written the focal length of a given convex lens by distance subject matter. What the value you got when you finding when you calculated when you find the distance subject method. When you find the focal length by distance subject method, you have to write it. And the focal length of the given convex lens by the methods which you used is you have to write, and then the focal length of the focal power of the given convex lens. If focal length is asked, you have to write, otherwise you no need to write the focal length, just you have to write whatever you were asked to determine or find out. So that is the writing of a result. So here the make sure that you have to write aim, apparatus, theory and formula. Simply don't derive a formula. Here you have to only write the formula. You have to write the formula and simply you have to mention the words. What are the terms involved in the formula? For example, f equals to u. For example, here f equals to uv by u plus v. F equals to uv by u plus v. Then simply where you have to write where f is a focal length, u is the distance between the object and the screen, and v is the distance between the screen and image. Simply you have to write the formula and terms involved in the formula. That is the way of writing formulas in the experiment. And theory involved. What are the theories involved? If any definitions are there, you have to write definitions. Otherwise, no need to explain all the things. And no need to describe the experimental setup. You have to write a procedure. So, how you are going to perform the experiment by using these apparatus. So you have to write the procedures. Then you have to perform experiment. By performing experiment, you will get the required values, required data you will get. Take the data, note down the data in the tabular form. Then you have to calculate the data by a proper procedure. So in this experiment, you will get a data that is u and v value. So you have to substitute the u and v value in the formula. So formula is f equals to u v by u plus v. And if question rises, if you are asked to plot a 1 by u 1 by v graph or determine the value of focal length and focal power from 1 by u 1 by v graph, then you need a 1 by u 1 by v values. To get the 1 by u 1 by v values, simply do the calculation 1 by u, whatever you have the value. So from that you will get a u and v values. So you will get a u and v values. Now substitute all these values in the formula and take the u 1 by u values on x axis, 1 by v values on y axis, then plot a graph. Now you will get some points. So by joining all these points, draw a line. It intersects at x axis and y axis. The distance from the origin to a intersecting point is taken as 1 by u, and another point on x axis y axis is taken as 1 by v. So from this you will get a values, and you have to substitute that values, that is f equals to 2 by OA plus OB. So here this is the OA and this is OB. You will get a f value. So you have to substitute the exact values, otherwise you will get a wrong answers. Then the question rises. So now you have to, you got the f value, right? So you have to calculate and you have to write the precautions, whatever the precautions you have taken while performing the experiment. So basically minimum you have to write 3 to 4 or 5 precautions, because the precautions has a marks weightage. If you are not writing any precaution, you are not following any precautions while doing experiment, it leads to damage the devices, damage the instruments, or damage for any problem. Health hazardous problems will occur in the laboratory. When you are performing any experiment, you have to make sure that you have to take all the precautions given by the instructor or given by your lecturer. Given instructions should be followed strictly, and you have to mention the minimum 3 to 5 precautions, because in our syllabus we are giving 3 marks of 2 to 3 marks of weightage for the precautions. Then finally you have to write the result. So result writing is more important. So for he is asking, the examiner is asked for the focal length only. You have to write the focal length, that is some number 25 or 20 or something, and you should write correct unit also. So half of the marks are allotted for the number, half of the marks are allotted for the unit. Otherwise you will lose your marks. And then after the completion of your experiment, there would be a viva voice. So what is the viva voice? The examiner will ask some questions on the experiment which you done in the laboratory. So based on theoretical questions or based on experimental questions, he will ask the questions. The viva has a more significance in the laboratory, because you are able to answer all the questions, answer effectively, then you will get a good marks. So here this is the procedure to find the Here the focal length you got, and focal power is a reciprocal of the focal length 1 by f. It is taken in the meters and the units are the diopters. So this is the procedure, and experiment to perform experiment to get the focal length and the focal power by using convex lens. This is the procedure and the experiment you have in our class, right? So by this, hope you get the clear idea how to perform experiment and what to write in the examination, or what to write in the experimental notes. In laboratory notes, you have to maintain a notes to write whatever you are doing work in the laboratory. So you get the clear idea what you have to write and how to perform a experiment. This is the first experiment in the second semester we have in our syllabus. So hope you got the clear idea regarding the determination of the focal length and focal power of a given convex lens by using UV method, 1 by U 1 by V method, or UV graph, 1 by U 1 by V graph. So question rises differently, different form of question will rise on this experiment, but one student should attempt only one question. One question will rises comes to one student only, not all the questions you have to perform in the examinations. So make sure that you are going to write only whatever he asked in the examination. Your examiner asked for find out the focal length of the given convex lens, but you are writing in aim as determination of the focal length and focal power of a given lens, then the examiner easily finds out you don't have any clarity on the experiment, right? So make sure that you have to follow the instructions, you have to take care all the precautions and perform the experiment. Thank you.